Hey, what's up guys? Thanks for tuning in once again. Now today we have a fairly interesting topic about dementia. Apparently a while ago there's been a vaccine being tested in laboratories for it and supposedly it proved successful on mice. Of course, any progress we have in medicine is somehow going to make someone unhappy. Say the anti-vaxxers. Of course they'd be outraged knowing that more vaccines are potentially going to be injected into people. This person we're responding to today has a small channel dedicated to bringing awareness to the horrible vaccines being injected into your children. And it's been a while since I've batted an eye towards small channels, so today we're gonna do some bullying. I'm only joking. Please, I'm not a bully. Anyway, before we begin, I must give a mention to today's sponsor. You guys already know who it is. That's right, NordVPN. They have supported me for a year now, and it's been absolutely great working with them. If you haven't heard of them already from my other videos, they're an encryption service that provides you with the means to connect to thousands of servers around the earth in over 60 countries. You can connect to multiple devices, and it's incredibly easy to use. Just use one button to connect. No matter if you are looking to protect yourself or if you want to watch shows unavailable to your country, NordVPN is the solution to all these problems. Just head over to nordvpn.org/stick using the code STICK for 70% off for 3 years, which gives you an extra month for free. That's nordvpn.org/stick, link in the description. Once again, thank you so much to NordVPN for supporting me throughout the year. In all honesty guys, their service is awesome and they've played such a big role in keeping this channel running. Without further ado, we shall get started on the video now. Oh, by the way, I've gotten incredibly busy, so I'm looking for more fan arts to show off in my videos. It's a way for you guys to show off your artwork, and it's good for me because it saves me editing time, since I spend all my days with my face buried in a textbook at the library now. Discord is the best way to send them to me if you're interested. I apologize if I respond late, just be patient with me. Anyway, on to the video.
Hey folks, thanks for joining me on experimental vaccines. I found this on Newsweek. Experimental Alzheimer's disease vaccine tested in mice cut dementia cases.
Ah, yes. Now, admittedly, I don't do a very good job at keeping up with the latest scientific discoveries. At one point I did, but now I hardly do. So some of you guys are definitely more aware than me on what's going on in the latest medical discoveries. God, I really need to keep up with my game. This Alzheimer's vaccine, if it were huge news, is something I really shouldn't have missed.
Now, first I'm curious, how did they know the mice had dementia? How did they figure that out?
Very good question. I don't have to read the original source to give you the answer to that one. Most likely there was a mouse line designed specifically with Alzheimer's. See, we know a lot about this disease already. We know some of the mechanisms, some of the proteins involved, and its pathology. In other words, we understand the biochemistry behind it. So if you were to purposely induce these conditions into mice, then it's a simulation of Alzheimer's. I'll get into more detail later. This can either be designed by researchers of this paper themselves, or it could have been done previously by another group of scientists, who then bred and passed down the traits of Alzheimer's to future mouse generations, which are then used by other scientists. This is actually very commonly done in other fields of research as well, such as cancer. And I'm sorry I'm always going back to cancer because that's just the field I'm most familiar with. There are cancer cell lines that get passed down that can be purchased, and also mice with or without certain genes. There's a whole list of them. It's cool. Research has gotten easier and easier over time. Now, just to be sure, let's look at the original source. Man, I hate looking for the source from secondary news articles. They always fucking hide it somewhere. It's so annoying. Anyway, good thing the original video has a link down below, which brought me to Newsweek. They had a link in the text which brought me to USA Today, and finally, they had a link that led to the original paper published by Alzheimer's Research and Therapy. So anyway, experimental vaccines. That's the name of his channel. You could have answered your own question just by clicking a few links like I did, because every research paper is required to have a method section where they detail how they conducted their experiment, and part of that means they have to mention how these mice had Alzheimer's. So let's take a look, shall we? Immune responses were monitored by ELISA and enzyme-linked immunospot. Brains from 20-month-old 3XTGAD mice that had received DNA amyloid beta 42 immunotherapy were compared with brains from age and gender matched transgenic amyloid beta 42 peptide immunized and controlled mice by histology, Western blot analysis, and ELISA. Protein kinase activation and kinase levels were studied in Western blots from mouse hemibrain lysates. Amyloid beta here is a driver for Alzheimer's. Immunotherapy is, the vaccine. Histology, Western blot, and ELISA are analyzing techniques which were done on mouse brains of treatment and control groups. So as you can see, they mentioned 3XTGAD mice, which is the mouse line they used. If you scroll down to the meat of the paper, they actually give a lot more detail on what this mouse line is and how they obtained them. Oh, Professor Stick, what are 3XTG mice and how do we know they're reliable? Well, that's easy. You can just find the original paper that details how they're created. I tracked it down to this paper, so let's just briefly read the abstract here to get an idea. The neuropathological correlates of Alzheimer's disease include amyloid beta plaques and neurofibrillary tangles. To study the interaction between amyloid beta and tau and their effect on synaptic function, we derived a triple transgenic model harboring PS1, APP, and tau transgenes. Rather than crossing independent lines, we microinjected 2 transgenes from single-celled embryos from homologous PS1 knock-in mice, generating mice with the same genetic background. 3xTGAD mice progressively developed plaques and tangles. Synaptic dysfunction, including LTP deficits, manifests in an age-related manner, but before plaques and tangle pathology. So yeah, you can kind of understand how these mice were created now. Here's the translation. They injected certain genes into the embryos of mice. These genes integrated into their DNA and works to generate PS1, amyloid precursor protein, and tau to encourage tangles and plaques, which is what actual Alzheimer's patients have in their brains. Okay, are we done? I spent way too long talking about how these scientists know that their mice had Alzheimer's, so let's progress into the next section, shall we?
Let's see what they say here. Researchers at the University of Texas Southwest Medical Center in Dallas said a new experimental Alzheimer's disease vaccine showed promising results during recent testing in mice and are hopeful that the vaccine will make it to human trials. In the animal trials, researchers said the experimental vaccine showed it could delay the effects of a degenerative brain disease. The journey from animal test to human is a long and arduous, and many promising cures do not withstand it. But a senior author of the research publication, published this week in the journal Alzheimer's Research and Therapy, told USA Today if the vaccine is proven safe and effective during human trials, it could reduce the total number of dementia diagnoses in half. If it's approved, and maybe in half. It's so crazy, the terminology they use here.
Crazy indeed, but that's just what secondary news articles do. They try to interpret things and present them in an entertaining way. In no way did the actual scientists make any claims about its human trials and what it could mean for society. Obviously, they want to get the immunotherapy through to human trials. That's like the goal of any medical researcher. But the truth of the matter is, and scientists are aware of this, is that many potential treatments simply don't get far. As sad as that sounds, it's true. Countless treatments have made it to human trials and fail there. It's an incredibly rigorous process, and for good reason. Chances are this vaccine probably won't pass. But then again, this article was written almost a year ago. Perhaps there has been some updates about it. I'm too lazy to look for it right now, though. Oh, come on, don't give me that face.
The experimental vaccine, could be a monumental push forward in the fight against dementia, with previous Alzheimer vaccines causing damaging side effects, including brain inflammation.
Ah, the magic word for people who deny modern medicine. Inflammation. What a scary word. Of course, if you're going to use an immune response, you'll have some degree of inflammation. If it's in the brain, inflammation can be bad. But that's going to differ depending on what type of vaccination this is. Just because previous attempts failed does not mean we give up.
So it's like, oh yeah, we've killed people trying this before, but we're gonna try it again.
What? How in the world did you interpret that as killing people? There are ways to handle inflammation. We don't just give an experimental treatment to people and when their brain inflates we just go like, oh well, looks like you're dead. No. Obviously you then treat those patients.
Here we go with recent tests on monkeys and rabbits, because we just look just like monkeys and rabbits, found the vaccine works by promoting the body to produce antibodies that reduce the buildup of amyloid and tau. Both proteins are typically indicative of the degenerative brain diseases present in the body.
Ah yes, the science of Alzheimer's. It's fascinating. Now, you guys probably have no idea about amyloid plaques and tau, so I'll give a brief lecture about it. Hopefully this will help you better understand what causes Alzheimer's. Now, Alzheimer's is very common. Almost 50% of people who are over the age of 85 have this disease. So what is it exactly? One of the mechanisms discovered was a protein called amyloid precursor protein, or APP, which we actually read in the papers earlier. Anyway, APP is cleaved by secretase enzymes into amyloid beta peptides, either 40 or 42, depending on the shape of APP to begin with. APP shape is determined by genetics, ones that have been passed down through generations. So yes, Alzheimer's has a degree of genetics to it. Anyway, if you have too much amyloid beta 42, then they aggregate into amyloid plaques, which is neurotoxic. But amyloid beta isn't the only problem involved. As you read from the article, there is also tau. When cells are diseased, they can release tau units, which aggregate to form tangles. This, in combination with amyloid beta 42, is what was discovered in 1963 by electron microscopes. So the hopes of many treatment ideas is to directly attack amyloid beta 42 and tau, such as through antibodies. An immune response towards these proteins could be the solution, which is why the idea of a vaccine was developed. That obviously caused many other problems, so clinical trials are crucial in making sure these treatments are effective and safe.
Know this, okay? The Alzheimer connection is really about aluminum in the brain.
Huh, funny. The studies and experiments done seem to be pointing at amyloid beta and tau, not aluminum. Weird, isn't it?
And how it deposits itself in this cholesterol called GABA.
Okay. Yeah, you can't say deposits itself in a cholesterol. What does that even mean? Cholesterol is just another type of molecule. Don't tell me you think aluminum can turn into cholesterol somehow. Second of all, GABA is an inhibitory neurotransmitter. It's not a cholesterol of aluminum or whatever you think it is. Aluminum, cholesterol, and GABA are such different things that they have almost no relation to each other.
And if you start to look into vaccines, most of them have aluminum as an adjuvant.
Yeah, it's just an adjuvant to trigger an immune response. That way you won't need to inject more than you need to. Yeah, maybe you want to demonize aluminum, but it's usually in a salt compound such as aluminum phosphate. This fear of aluminum is unjustified. There's nothing wrong with that amount of aluminum being injected into your body because you get more of it just by eating food.
And deodorants have a lot of aluminum in them. The chemtrails that they're spraying on us have a lot of aluminum in them.
Ah, yes, the chemtrails. Can't go a single video without mention of that now, can we? Anyway, guys, that's the end of this video. It's been refreshing to talk about molecular biology once in a while. I hope you learned something new today, and if you did, be sure to subscribe to, learn more, I suppose. Thank you to Mark Orsini and Fireshard for your loyal support over at Patreon. We will be back next week for another awesome video. Take care and don't breathe in the aluminum. Also, don't forget to send me fan arts.